Okay, so chapter 9 is microbial genetics, and it focuses on introducing us to DNA again, and genes and how they make proteins, and then genetic engineering, which is when we purposefully mess around with the genetics of microbes, for example. Okay, so we will look at DNA and the chromosomal DNA compared to the plasmid DNA. And we're going to look at mutations and the changes that they can cause. And then the three forms of gene recombination that can lead to new chromosomes or plasmids in a microbe. And then we'll look at the basics of genetic engineering just a little bit and talk about what that can mean for microbes and us. Okay, so bacterial DNA there are two types, okay? So we're focusing on bacteria pretty much this whole section because bacteria have special genetics that are really interesting and unique. So anyway, so bacteria, have both chromosomes and plasmids, although only the main chromosome is the essential piece. Okay, whoops, too fast. So the bacterial chromosome is highly compacted and folded up into this kind of pretty little flower shape. And so it is held in place by these little loop anchors that are just like a little molecule that holds the DNA into this flower shape. And that allows it to be super condensed inside of the bacterial cell. This picture on the right, they're a little hard to see, but all those little squiggly lines in the back, that is exploded bacterial DNA. That's how much can come out of one bacterial cell. So you can imagine how super condensed it is to stay in the nucleoid of the bacterial cell. So like I said, the one thing about the chromosome is that it is absolutely essential. Okay, here on the left we can see bacterial DNA, and on the right we can see plasmids. And the plasmids are non-essential. They just code for other specialized things. So it's almost like getting little superpowers on those plasmids, but you don't have to have any of them to survive. That's what the bacterial DNA is for. Okay, so we should understand that already. I had mentioned that in previous lectures, but definitely read about that in the book if you're still not sure about the differences between the bacterial DNA and the plasmids. Okay, let's focus just a little bit longer on the bacterial DNA though. So the genome is all the genetic information in a cell. And so, for example, E. coli has 4,300 genes on its one circular main chromosomal DNA. But the number of genes can vary quite widely. So here is two extremes. The smallest known microbial genome is a Mycoplasma genus. And this one species has 580,000 base pairs, only encoding for 525 genes. That's not very many, 525 compared to the 4,000 in E. coli. But then some of the largest genomes of bacteria occur in the cyanobacteria, those photosynthetic bacteria, and they have 12,000, more than 12,000 genes. So quite a bit more than E. coli. So it can vary quite a bit. And we have more than that, but you know, we're not microbes, and we're focusing on microbes. So this is what it looks like if we map out the entire genome of a bacteria. This is represented in a circle, remember, because bacterial DNA is in a circle. And so they've literally mapped out every single base pair. That's what all the little colored pieces are. And then they are showing us in here some of those genes and what they do. Nothing that you have to memorize, of course, but it's just interesting to see how they have mapped all that out and they know exactly what all those genes do, for the most part anyway. So there's still some secrets in there, but you can see how detailed this is. Staphylococcus aureus is just a very common research bacteria and commonly found on the skin too. Very common bacteria that we've researched heavily. All right, but now let's talk a little bit more about plasmids. Those are the small little circular pieces, again, not essential, but they may help the bacteria in various ways. So, for example, they may carry virulence factors, which would make us more or less, probably more sick if they're a virulence factor. Or they might make them antibiotic resistant. That's a very useful plasmid. But it could be just simple things like helping them make a new enzyme that helps them metabolize something a little bit differently. So they're not all fancy, like making them more contagious to us or making us sicker or being antibiotic resistant. They're not always such special powers, but they can be pretty simplistic plasmids and still be very beneficial to that little bacteria. So these may have played a very important evolutionary pathway in bacteria because those plasmids can move around from one species to another. And so you may have a bacteria that was not previously pathogenic to humans, and then it gets a plasmid and now. So as an example, E. coli didn't used to be pathogenic to us. It wasn't until it mutated that a strain evolved to be pathogenic to humans. All right, so let's talk about mutations in general. That's going to be one of the topics for today. So mutations are a change, a permanent change, in an organism's DNA. Okay, so if we're talking about a bacteria, an individual bacteria, then that one bacteria can have a mutation, and then it's going to be passed on to all of its future progeny, which means when that one cell divides into two, the DNA is going to be replicated through that process we talked about last time, and then that same loop of DNA will be in each of the resulting bacteria. Okay, so if one bacteria has it, then when it divides in half, both of those bacteria are going to have the mutation as well. And they can be good changes, bad changes, or have no effect. Okay, just kind of depends on the mutation and how it reads. And I'll give you an example of that in just a second. But it could be something great like an antibiotic resistance, which of course is not great for us, but it's great for the bacteria. It allows the bacteria to live. Okay, so that's how big of a change this can be, although oftentimes it might be something unnoticeable or even harmful to the bacteria, like cancer is to us. When our cells mutate, we get cancer. And so, it could be something just as catastrophic to a bacteria when it mutates. But what's also important to understand about mutations is just because the DNA changes, it doesn't always mean that there's going to be a resulting change. So a phenotype is an observable characteristic. So for example, I have the genetics of brown eyes and blue eyes because my mom has blue eyes. So if you know much about genetics, I have the big B, little b combination. But only the big B shows because that is the dominant characteristic. So brown eyes is my observable characteristic, my phenotype. Okay, so for bacteria, they don't have brown eyes, obviously. What do they have? They have their cilia or their flagella or their antibiotic resistance or the type of enzyme pathway that they use to metabolize sugars. All different things can be phenotypes, but it's just the observable characteristics, even if there's something hidden in their genetics that is slightly different. Okay? So we call their natural state their wild type genome. Okay, that characteristic, or phenotype, excuse me, the wild type phenotype. And when it mutates, then we can call that individual a mutant. Okay, so for example, if there is a strain of E. coli and one of those E. coli develop a mutation that allows them to be resistant to a certain antibiotic, then we would call that strain mutant strain because it's not like the wild strain that we are used to seeing. Okay, so one of the types of mutations that we need to talk about is a point mutation. And that's exactly what it sounds like. It's one point on the genome that has changed. Okay. Okay, so this is a single nucleotide change, and so in other words, only one letter of a base pair is changed. Okay, so if we look at this, I'll bring it up larger here so we can look at these examples. So this is our original DNA, and we're just looking at these three little base pairs at a time. So uracil, uracil, adenine. Okay. And that is going to give the protein or the amino acid piece leucine, okay? And that will form a protein correctly or not. Okay, so let's assume that UUA is the proper amino acid that we want to put into this protein to make it the correct protein. Okay, but then let's say something happens and this one has mutated. So we're going to look at 4 different examples of mutations and the result of those. Okay, so let's say, for example, the mutation was a frame shift mutation that added this extra G in here. So instead of UUA, we have UUGA. Okay, that is a really bad mutation because we've put an entirely new letter in. We've shifted all the other ones down and stuck a new one in. So that's called a frame shift. Okay, because all the letters that come after that are going to be a little bit wrong because we've kind of shifted them down. What is really essential to this conversation, by the way, is that you remember that when we make proteins, we have to do it in 3 base pair sets. Okay, so you can see what should have been UUA is now going to be read as UUG, and the next one is going to be A whatever. So we've shifted everything. So that one is definitely going to make a big change in that resulting protein that is made. The next one is a silent mutation. And the silent mutation is one in which the base pair is swapped for the wrong one. So instead of UUA, we have CUA. But coincidentally, CUA also codes for leucine. Remember this? That's our original one that we named up here, that the UUA is coding for is leucine. And in this silent mutation, there was a mutation, but it's silent because we still get the same result. Both of those codons, both of those sets of 3 make leucine. Okay, so no problem there. The next one, though, is a missense mutation. And that one is going to be a changed amino acid. So in this example, we have GUA instead of UUA. And that is going to instead code for valine, which is obviously not leucine. So that's a different amino acid. This has caused a mutation that will be evident in the bacteria. The last one then is called a nonsense mutation. And this is one in which you've swapped things around. So instead of UUA, we have UAA. So we've just switched that middle one. And coincidentally, this one is called a stop codon. So it comes to this, then there are just certain ones that are stop codons and start codons. But when it's trying to make a protein, it's going to read this and say, oh, this is the last one we should make. And nothing else will be made after that, even if there is more messenger RNA after this. So that is also a pretty big problem. So obviously the one that is the least problem, the least likely to cause an effect is that silent mutation, followed by the missense mutation that's just going to put one wrong amino acid in there. And then the frame shift and nonsense are both really bad mutations because they're going to totally change what comes after them. For the rest of that protein will be entirely wrong. Okay, this last little one here was just going over the details of that frame shift mutation and that it will, the other page dealt with the point mutation. So a separate page for that one. But we'll keep going. All right, so what causes mutations? Sometimes they just happen. That's our first one there, that spontaneous DNA replication error. When the DNA is being replicated, it just makes a mistake. And we actually have molecules that go through and look for those mistakes, but sometimes they miss them. So it could just be that kind of an issue that just happens randomly, and the molecule that fixes it didn't catch it. Okay, but we can also have exposure to things that cause mutations, like UV light or certain chemicals that can, what they essentially do is literally go in and disrupt the DNA itself. Okay, so in the first example in this picture, we see that it's ultraviolet light, and it has kind of messed up our little structure of DNA here and made it off-center. So instead of it fixing and bonding correctly, it bonds incorrectly. And then we can have a new base pair slip in there instead. Okay, so mutagens are things that cause mutations. All right, so now we're coming to our third major topic here, genetic recombination. And this is how how bacteria can share their chromosomes. Okay, And so what we have to understand, again, about bacteria is that they undergo binary fission. And so the, quote, offspring look and are genetically identical to the parent cell, right? This first cell here just literally doubles its DNA and then splits in half to make these next two cells, right? And then those split in half to make their next two cells. So if you have a little dot of bacteria on a petri dish growing, that little dot is, thousands and thousands of bacteria, and they are all genetically identical to each other, okay? Unless a random mutation has occurred, but we can expect them to all be identical. So how do we get any diversity in bacteria if they're just dividing like this? Shouldn't every bacteria be exactly identical? We never have new ones? No, because of this genetic recombination. So let's talk about the types of genetic recombination. All right, the first one is called horizontal gene transfer. And what that means, it's a fancy word for just saying the genes from one bacteria somehow make it into another bacteria. So do you see what I mean by horizontal? It means it's going from one to another, not through the process of having offspring. Okay, so the way I gave my genes to my children was that I gave birth to them. They have part of my DNA and part of my husband's DNA, and that's how we made that child. But imagine if humans could do horizontal gene transfer. That would mean that I would give some of my chromosomes to somebody else. Okay, and that's not how that works. But that is how it works in bacteria, because they do binary fission, so they have to have some way to share their genetics. Okay, so there are 3 ways that we can do that, and they're listed here, but we'll look at each one on their own page. So one is conjugation, where the DNA is passed from one cell to the other directly. Transformation, where it is just DNA that is loose in the environment and it gets picked up by another cell. And then transduction, when a virus picks it up and moves it over. So in all 3 of those, the result is that we have moved this little specialized piece here, this little green piece, into the new cell here, into the new cell here, into the new cell here, so that we have the recipient cell being now with that little piece of DNA inserted into it. All right, so let's look at those 3, starting with conjugation. Okay, so this is conjugation, and it's not important for me that you know all these details over here, but I do want you just to be able to explain that we have a conjugation pilus that is going from one microbe to the other. And when that happens, we make this little bridge, okay? So we start with a pilus, and then it forms a bridge, and then the DNA comes through, okay? When the DNA comes through, now the new cell has that DNA. Okay, so this is a type of sexual sharing of genetic material, right? We are taking the plasmid from one, replicating it, and sending the second one there. So you can see that I kind of skipped that conversation here, sorry. Here is where that plasmid is replicated so that the new cell gets it here, and the old cell still has it as well, okay? All right, so that is conjugation, the sharing of a plasmid through a conjugation pilus. The next type is transduction, and transduction is the viral method. So a bacteriophage, which is a virus that infects a bacteria, will insert, as you can see here, at the top, will insert its DNA into the cell as it normally does, and the cell does what it does and make more of the DNA. Okay, but then if we go down here, we see that, this one in blue picked up some of the bacterial cell's DNA. Rather than the viral DNA like it's supposed to, the virus picked up the bacterial DNA on accident. Okay, not that it meant to do that, but sometimes when things are getting repackaged, that just happens. Okay, now when those little viruses explode out of the cell, that virus can go around and inject into another bacteria. Here's its DNA injecting in this letter G picture. And then it can insert itself into that second bacteria's DNA. So they're representing it by that little B. See the little, so they're trying to show in this picture up here, we have like A, B, and D, these little sections here. And then B happened to get into this virus, and then we follow the B all the way through, and now it's in the new bacteria. Okay? So again, bacteriophage transporting some bacterial DNA into a new bacteria. The last type then is transformation. And this is when DNA is just in the environment from a different bacteria that died maybe. And you know, you saw in that picture, let's see, go back there real fast. In this picture on the far right, that DNA just splattered all over the place when the bacteria died. So you can imagine if there was a bacteria nearby, it'd be very easy to absorb some of that DNA. So that's what's happening with the transformation. Okay, so there's this little DNA fragment that's in the environment for some reason. It's picked up by the recipient cell, and then it becomes part of that bacteria's DNA. That's it. Okay, it can just insert itself into the other chromosomal loop. Okay, there's one other that we didn't talk about on the previous page because it's not a horizontal transfer. This is different. This is a transposon. So transposons are a little bit of a different type of horizontal transfer from, one way to think about it is that within the initial bacteria, the little segment that we're talking about has to hop off of the main chromosomal part and onto a plasmid, and then the horizontal transfer can occur through one of those other methods. Okay, so let's walk through this. So up here at the top, we have that blue main chromosomal DNA with a little black piece that they're using to represent the transposon, the jumping part. And then this bacteria also has a plasmid. And so the little black part here is just going to hop over onto the plasmid, and then we see that here. Okay, now through one of those methods of conjugation, transduction, or transformation, this plasmid can be transferred to another cell. That's the horizontal part. That's the horizontal transfer now. And now the new cell has that, and it can then hop back on to the new cell's main chromosomal DNA. Okay, so we call those jumping genes, and it allows a piece of a chromosomal DNA to hop onto a plasmid, go to a new cell, and hop off the plasmid and attach to the second cell's chromosomal DNA. Crazy, right? So we can see that bacteria, even though they have this very simple method of binary fission as their way of making more of themselves, they have these crazy ways that their genetics can get mixed up and improved, hopefully, for the bacteria's sake, and help them survive better. Okay, so we also have some negative side effects for us in this process as well. So with genetic recombination, those methods that we already shared, we have this issue of antibiotic resistance. And this is a really big problem for us because we don't actually have that many antibiotics. I mean, it's a pretty long list, but as far as bacteria are concerned, they're beating every one of them, slowly, but they are. So the way that this can happen is like such. I'll explain a scenario in which this could happen. Let's say, for example, you have an infection. Maybe it's a staphylococcus infection, and it's making you sick, and so you start to take an antibiotic. And the antibiotic is going to kill off some of those bacteria. But then you have a situation where there's another bacteria inside you, and as some of those pathogenic bacteria are starting to die off, some of the DNA is picked up by this other strain, maybe an E. coli strain. And now the E. coli strain, let's say it could pick up that same pathogenic gene and make you sick in the same way. Well, the exact same thing can happen with antibiotic resistance, even within the same species. So let's just look at Staph aureus, for example. Let's say that you have the wild type of Staph aureus in you, And then one of them has a mutation, and now it's a mutagenic strain. And then you're taking an antibiotic to kill the Staph aureus. But that mutation now is going to protect that bacteria. So while you might be killing off the wild type, you're not killing off the mutated type. And so now that mutated type can reproduce like crazy. The other bacteria is being killed by the antibiotic, and so it has less competition. And so that is the way that we get a lot of the antibiotic resistance. And of course, it can also be transferred to other bacteria then, which is doubly bad. So that really threatens the prevention and treatment of our bacterial infections that we have. And so there are some strains of bacteria that are resistant to everything, every antibiotic that we have, except for maybe one. Okay, so they will not let anybody take that antibiotic for that bacteria unless they're otherwise going to die, because we don't want to allow that bacteria that is resistant to everything else come into contact with this one type of antibiotic that still works against it. And that is happening more and more commonly, that we have to keep adding medications to the list that are no longer effective against certain types of bacteria. Okay. All right, so there are some examples there, not that you need to know those names, and we will be going through more of those as we go forward. But what if we want to do recombination on purpose? What if scientists want to mess with the DNA of bacteria on purpose? Then we call it genetic engineering. Okay, so genetic engineering is not a natural process. Gene recombination is, but when scientists do it on purpose, we call it genetic engineering. So we're just manipulating the genes of a cell or an organism using high-level technology to do that. And this is how we do it. It's kind of crazy. So we take this strand of DNA that you see up here at the top, and we slice it in a very certain way. And it's going to be sliced in this jig-jaggedy direction like this, so that we kind of open it up like you see here on the right below this, where the ends are exposed. So these base pairs don't like to be exposed like this. This makes them feel kind of naked and empty. And they, as soon as they are introduced to other base pairs, they will automatically hook up. It's just a chemical reaction. So we call those sticky ends. Okay, so a restriction endonuclease, which is just a fancy word for the enzyme that does this, will slice it in that certain way to make these sticky ends. And then we can start purposefully sticking stuff to those sticky ends. Okay, so we see that in this bottom picture here. And it's showing us that we can split in a certain way and then rehook just the way that we want to. Okay, so they know where these codes are. They know which enzyme is going to work to split it in exactly the right spot. So they can, it's just like working with a pair of fine scissors, and they can pick the exact location where they want to open up the bacterial DNA and stick something else in there. Okay, this is what we're looking at here. So this was our original E. coli plasmid here, and this is what we want to put into it, a piece of a virus's DNA. So we will open up the bacterial DNA in the gray, creating these sticky ends here and here, and then put the viral DNA, just click it into place, and it will create a whole new bacteria that now has this viral DNA in it. So we use this for all sorts of crazy things. Like we can have bacteria make antibiotics for us. We can have them make insulin for us. We can have them make growth hormone for us. Lots of different examples of the things that we can make with this intentional gene recombination, otherwise known as genetic engineering. Okay, and we will be looking at more examples of those as we go through, but this is the genetics of how that works. So when we do it in the future, you'll already understand the genetics of it, and we can just talk about all the cool stuff that we can do with that. All right, well, that's it for this chapter. It's a heavy chapter, so make sure that you read the book on this chapter. It's pretty interesting stuff, and it will definitely help you as we move forward if you learn this stuff well now.